Institute, Cuttack, and he has areas of specialization is CRISPR-Cas9 tools, development, and precise genome editing, base editing, genetic engineering, transgenics, also the very important one, host pathogen interaction. Has done his PhD and currently is working at Crop Improvement Division, Cuttack, and previously was a Fulbright visiting scholar in the Pennsylvania State University, and he was also a scientist, worked as a NBPGR, Division of Genomic Resources, and he has 20 research papers, 3 review papers, book chapters and all are there, and he was awarded several awards of recognition, awarded at EA Science Program for Excellence in Science, Fulbright Fellow, Postdoctoral Fellowship, Jawaharlal Nehru Award for Outstanding Doctoral Thesis, Young Scientist Award and International Travel Grant. He has got winner of 2 Outreach Lecture Fund, winner of Annual Postdoc Research Exhibition Award, and Early Career Research Award and Merit Scholarships. He was there, and this is in brief the biodata of Dr. Molaji. Now, if Molaji is convenient, he can. Can I request Molaji to go on air?
Yeah, sure. Okay, thank you very much for inviting me, and it's wonderful to be here. You know, I can see there is 1120 participants already joined to this webinar, and it's really exciting to share some of my research experience and also some others research what is going on in CRISPR-Cas fields. So yeah, and also sorry from the I apologize to the participants on behalf of the organizing committee because of this technical problem. So I was supposed to give the second lecture, but because of some technical problems I am compelled to give the first lecture. So let me share my screen. Yeah, is it visible now?
Yes, yeah. It is visible, Molaji.
Okay, so welcome again to all participants to this nice initiative of a one-day webinar from ANGRAU, Hyderabad. And it's my real privilege to be here to present the first lecture on CRISPR-Cas tools. What are the different tools derived from CRISPR-Cas technology and how it is impacting crop biotechnological innovation, including basic plant biology and also applied biotechnology. So yeah, I will give you a brief about different kind of tools that are available with us and what kind of engineering we could do with those tools, and I would give you a few nice examples to better understand how it is impacting the current biotechnology. So let me give you a brief snapshot of outline of my today's presentation. And the first part, I would be talking about what is genome editing and a brief introduction of CRISPR-Cas9 technology and how it really works and how it could do the genome editing application. And then in part 2, I would be going beyond the normal or conventional CRISPR-Cas9 to different tools and techniques and citing nice case studies for each of the tools and techniques that I am yet to discuss. So if I go in details of part 2, I would be considering discussing gene regulation and then epigenome editing and then CRISPR detection. And then I'll be talking about base editing, and at the last, the most exciting one, just recently innovated, that is prime editing. So let me introduce, I always love to introduce my slides with this brush of our basic genetic understanding of DNA and gene, how they are. So, human body is a higher eukaryotes like the rice plants and all other eukaryotic organisms. They are nothing but mass of trillion of cells. So they are made up of trillion of cells. So here's an example of plant cell. So and this cell, these are the different parts of the plant cell, and it has a nucleus, and this nucleus is the main function. This is called the head of the cell because it contains the genetic material in the form of chromosome. So chromosome is nothing but a thread of DNA that is wrapped around the histone protein. So the chromosome is formed by DNA and histone protein. So keep in mind here, I'm going to discuss about DNA. So forget about histone protein. So DNA is a long thread, and it is the genetic material of most of the higher eukaryotes. If you see the whole DNA thread, the entire DNA of an organism are not important according to the current knowledge. Because some part of the DNA is functional and other parts are not functional. So this functional part highlighted here is called the gene. So if you want to define a gene, you could simply define like it is a functional unit of DNA. It's called gene. So why it is a functional unit? Because this portion of DNA, it actually expresses a character. Because this DNA, after transcription, it produces RNA, and then after translation, it produces protein. So this transcription and translation produces protein from a single gene. So now, these protein actually govern a character for an organism. So if you see, our DNA has 4 different kind of letters. It's nothing but the combination of 4 different kind of letters, that is A, G, C, and T: adenine, guanine, cytosine, and thymine. So these sequence actually make up our genetic code, and this genetic code actually governs almost everything in an organism. So DNA is nothing but a sequence of A, G, C, T letter, and the different sequences makes the different gene. Nothing more than that. Just keep in mind. Now, if you see this slide, this is a very classical example of the problem with wheat, rye, and barley, because these food crop contains a protein which is called gluten, and certain group of people are not able to digest that gluten because they are gluten intolerant. So this is the problem in many Western countries. So why this gluten actually causes problem? If you see our small intestine, this is the normal villi in the small intestine, and they actually function to absorb nutrition from the food. So now, the problem with gluten is a protein, and it has 2 different kind of protein that makes the gluten. One is gliadin and another is glutenin. So this gliadin is actually recognized as an antigen by the body of certain people. So if gliadin, the body recognizes gliadin as an antigen, so they develop immune response against this gliadin. So now, because of this immune response development, so the villi became this kind of destruction of villi causes, and it looks like this one. So the people who are suffering from celiac disease, and this disease is called celiac disease. So if you intake a lot of gluten containing foods like wheat, rye, and barley, then you may have this disease. So the villi is distracted, and if you have a distracted villi, then you would no longer be able to absorb nutrition as you are supposed to do with a normal villi. So this is the problem with gluten containing foods. So you see, I told you that these gliadin actually are antigenic in certain people, and they develop immune response, and they causes this kind of destruction of villi, and because of this destruction, the absorption of nutrition became reduced, and you develop celiac disease, which have a lot of consequences. So this is a problem in certain groups. So now, because in my earlier slide I told you that one protein is produced from certain DNA sequence or certain gene. So now the question is, can we specifically deactivate the gliadin forming gene in wheat? So you could hypothesize that if you could deactivate gliadin forming gene, so you could develop a wheat that would have very less amount of gliadin, so the intolerant people can have the wheat without any problem. So the question here, I have to have a tool to specifically deactivate that special portion of DNA that is producing gliadin. So now, so suppose if you say this is a snapshot of DNA, suppose this portion of DNA is actually forming the gliadin gene, gliadin protein. So if you can block that gene, and if you can reduce the function of that gene, or if you can deactivate the gene, then you could get rid of the problem of more gliadin formation in wheat, so you could reduce the gluten content of the wheat. So is there a way to specifically deactivate this kind of gene which is causing a problem? So the way is genome editing, because genome editing is nothing but, I told you that genome is nothing but DNA, and DNA is nothing but the sequence of letters. So now one gene causes one kind of sequence, another gene has different kind of sequence. Now if you can change that sequence a bit, then you can deactivate a portion of the genome or a particular gene. So suppose you have written some report or something, project report or a thesis or a paper, and then you give it to your supervisor, to your sir, to edit it. So suppose you have done a mistake in something, in some one letter spelling is mistake, then your supervisor would correct it, would edit it. So same kind of thing. Suppose our genome contains some, maybe our genomes in crop genome, some kind of deleterious or some bad gene is there. So you could get rid of that gene. So you could edit the sequence of that gene, and you could get rid of their product. Okay, so this is the genome editing, nothing more than that. So now, if you see the tools available for genome editing, there are 4 different tools. If you see the historical perspective of genome editing, the first one is meganuclease, and then ZFN, zinc finger nuclease, and then TALEN, and then comes CRISPR-Cas9. So it is recently developed, just 7 or 8 years ago. So if you see this earlier 3, that is meganuclease, ZFN, and TALEN, they all are difficult to use, and it needs very expert protein engineering laboratory to work on these, because these other 3, meganuclease, ZFN, and TALEN, they depend on protein DNA interaction. To edit a DNA, to edit a gene, you have to design a new protein each time. Suppose if you want to go for gene X, then you have to design protein X. Then if you want to go to gene Y, you have to design another new protein, gene Y. Protein Y, to edit that gene. But in case of CRISPR-Cas9, it is the most easiest one among these 4, and it is the most economical one and most easiest to work with, because it depends on RNA and DNA interaction, not on protein and DNA interaction. So it depends on normal Watson-Crick base pairing between RNA and DNA. So if you want to shift from one gene editing to another gene editing, you have to design a new RNA, small new RNA. So you don't have to go for a new protein designing or protein engineering. So for one, suppose gene X, you have to design RNA X. For gene Y, you have to design RNA Y. Nothing more than that. And other components are fixed. So this is pretty easy to work with. So yeah, let me share with you this 2 figure. This TALEN was actually pioneered by Daniel Poydels from University of Minnesota. So I was privileged to meet him during a lecture. And then in CRISPR-Cas9, this Jennifer Doudna was the pioneer, and I met her in Cold Spring Harbor Laboratory in New York. So now go through the CRISPR-Cas9 technology. These slides will briefly introduce. I will introduce briefly how CRISPR-Cas9 actually works in a very simplistic way. So if you consider CRISPR-Cas9 system, you can call it as a scissor. Is a genetic scissor. You know that restriction enzyme can also called as a genetic scissor because it can cut a DNA for a special sequence, but this is a little bit different. So it has a two-component system. One is a small RNA that is called guide RNA that is customizable. So you could design your own RNA. And another part of this system is a protein that is DNA cutting protein, and that called Cas protein, that is CRISPR associated nuclease. So this is it can chop DNA. So if you see these RNA, this is called a single guide RNA, but it is actually derived from two different RNA. I am not going into details. One is CRISPR RNA, another is transactivating CRISPR RNA, and they are fused to form this single guide RNA. So this single guide RNA, and this is the Cas9 protein. These are the two components. Now, if I would like to give you an analogy of this system, so I could just call this Cas9 as a postman having two scissors, and this RNA you could call it as a genomic address. So now your target, I'm talking about genome editing, I'm talking about inactivating a particular gene. So the genome is huge, okay? And if you see rice genome, it's huge. So actually, if you want to make a specific edit to a specific DNA or a specific gene, then you have to go exactly at that position, okay? So this guide RNA is actually act as an address. So if you give this address, this is the genomic address. If you give this address, this post-Cas9 take the address, and it forms a complex. So this form is called, this is called CRISPR-Cas9 complex. This RNA is, suppose, guide RNA Cas9 complex, okay? So this guide RNA Cas9 complex, when they form complexes, they go to the genome and they find for similarities with the genome. What kind of similarities? You know, the RNA is also made up of nucleotides, okay? So they find similarities exactly. If you see, this is the RNA, and this is 5' and this is 3' end. So this 5' end 20 base pair sequence is very important in this guide RNA. So they find similarities with the 20 base pair RNA to the genomic DNA. So if they go to the genomic DNA and they search for similarities, if they find similarities with the guide RNA, then they bind. Whenever there is an exact match between the RNA and DNA, this is the guide RNA and this is the genomic DNA. So now whenever there is exact match, this RNA and DNA binds and they form a DNA-RNA hybrid. And whenever there is a perfect match, then Cas9 gets a signal that this is the position where I need to make a cut. So this is the perfect match is the signal for Cas9 to make a cut at that position. So you designed your guide RNA, and now Cas9 takes it and go and search in the genomic DNA, and if there is a perfect match, Cas9 gets the signal, yes, is it, so I have to make a cut. So now, then they make a double strand break in the genome. So this is DNA is a double strand. So one scissor cuts one strand, another scissor cuts another strand. So, you know, now it is simple to understand that the DNA is a whole function. Okay, now, if you make a double cut, then this gene is no longer functional. So you cut the gene and then you deactivate the gene, okay? So this is how CRISPR-Cas9 actually causes deactivation. Now, if you see what are the steps in this CRISPR-Cas9 mediated double-strand break and genome editing. The first step is, I have not introduced to you with NGG-PAM. So could you please mute? Could you please mute? Yes, thank you. Okay, and the first step, I have not introduced with this PAM. What is a PAM? PAM is a protospacer adjacent motif. So as this CRISPR-Cas9 system actually derived from bacterial adaptive immune system, it has been from Streptococcus pyogenes. So they need a protospacer adjacent motif. Suppose, in order to simply understand it, suppose you want to target, this is a genomic sequence, and if you want to target these 20 base pair with red highlighted one, so this is, suppose this 20 base pair you want to target, you want to make a cut here, but your target has to have PAM followed the target sequence. So this is the target sequence. Now it should be followed. Please mute yourself, all the participants. And okay, thank you. Okay, so what I was talking about. So suppose this is a genomic sequence, genomic DNA sequence. Now this red basis is the target that you want to make a cut in that sequence. Now this is your 20 base pair genomic target, you want to make a cut there. So now, but this could not be targeted until and unless it has a following PAM sequence. So if you see, this is a protospacer and this is called protospacer adjacent motif. So here in case of Sp-Cas9, Sp means Streptococcus pyogenes Cas9. So the Cas9 protein, which comes from Streptococcus pyogenes bacteria, is called Sp-Cas9. There are some other Cas9 also, like Sa-Cas9, Cj-Cas9, NME2-Cas9, all other kind of Cas9s. But for Sp-Cas9, the PAM sequence is a 3 nucleotide. One is N, then G, and G. N means any nucleotide, means it could be A, T, G, C. And GG means guanine. So you can see here, CGG means NGG. NGG is a PAM, and it is a prerequisite in order to be targeted that genomic loci. So I think you could understand from this explanation. This is PAM, protospacer adjacent motif. So in the first step, what they do, this Cas9 and guide RNA complex, they go and they search for presence of any PAM, means NGG sequence. If they find a PAM, they halt there. Oh, so this could be one of my target. So they halt there, and then in the next step, they form a R loop. What is R loop? R loop means when they find a PAM, they bind there and they denature the double-stranded DNA into single-stranded DNA locally. Locally means where they bound, because this Cas9 protein is actually has a special function that is called helicase, and due to this helicase function, it could denature or it could separate two, because DNA is a double strand, okay? So it could separate double strand into single strand. You could see here, this double-stranded genomic DNA is separated into two local separation. This is called R loop. And after this R loop formation, so now DNA is single-stranded. So now this guide RNA, this sgRNA, single guide RNA, they search for homology with this single-stranded genomic DNA. If they find homologous sequence, they perfectly match, then they bind there. And whenever there is a perfect match with the 5' end of the single guide RNA, then the Cas9 gets that signal that I already told you. So whenever it gets signal, then it makes a cut, double strand break in that region. So these are the 4 steps has to be completed in order to make a double strand break in the gene that you want to target. So till now it is clear that how Cas9 and guide RNA complex actually makes a double strand break. That's clear. Now how it could actually help in editing DNA. So how it helps in changing DNA sequences. So suppose this is clear to you that it makes a double strand break. So whenever there is a double strand break, now cell takes it very seriously, because DNA is a very important material for them, because it is a genetic material. So any kind of damage or any kind of break, any kind of cut in that DNA is not tolerated. So cell is very serious. So it all promptly activates the cellular repair system to repair the damage, to repair the break. So I would give you a very nice video how it actually repairs it. In my later slide, I have a nice video. So you see, double strand break has happened. Now cellular repair machineries are activated now in order to repair this one. So now the repair, there are 2 kinds of repair mechanisms. One is NHEJ and another is HDR. NHEJ is non-homologous enjoining, and other one is homology directed repair. So, but among these 2 repair in normal higher eukaryotes, this NHEJ is a predominant repair system. So it happens more than the other one. Okay, so this NHEJ has a problem, you know. It obviously tried to repair perfectly, because whenever the genomic DNA is perfectly repaired, then there is Cas9 and guide RNA complex is there. So it again binds and again makes a cut. Again, NHEJ repairs it. So repeated cutting and repairing, it actually causes the NHEJ to make some error in the procedure. So whenever it makes an error, erroneously, it introduces one or two new nucleotide in that cut position, or delete one or two or few nucleotides from that cut position. So this red one is actually signifies the random insertion or deletion. That is called random indel. And because of this random insertion and deletion in that gene sequence, the sequence what was earlier originally, it is disrupted. So the open reading frame of that gene is disrupted. So the normal protein that should be produced from that gene is no longer going to be produced because of this error-prone repair system. Because it already inserted one or two bases or deleted some bases, so the sequence is hampered and now the gene is non-functional. So how it works in gene knockout. And this other pathway is homology-directed repair. If I want to explain you very simply, so suppose there is a double-strand break. Now, if the cell want to go towards the homology-directed repair, I told you that it is a very, very less time cell goes through this one, especially during meiotic recombination. This kind of repair happens, you know, during crossing over. So, if there is a break in the genomic DNA, then cell looks for sister chromatid. I know that any diploid organisms, you all know that in any diploid organism, every single gene has two copies. Okay, so suppose this gene has another copy, so one copy is broken. So now, in order to repair through HDR system, it will see the other copy which is intact, that is in the sister chromatid. So, and this intact gene has the same sequence. So it will take that intact gene as a template to repair perfectly to the damaged one. Okay, so now this is the normal cases that cell operates. Now, if you want to go through HDR artificial, then you have to supply extra DNA that looks like the sister chromatid, but you have your intended change here, the blue one, that you want to make a change. Okay, so this is transversion or translation. So here, if you change, suppose mutation or any kind of single base pair or deletion, small deletion, you have to put in the middle, and the both ends should be flanked with homologous sequences, and the cell will be actually confused to take this one as a donor template, and it will copy this sequence into the broken part. So your intended change would be incorporated into the genomic sequences. Okay, so this is HDR, homology directed repair. So now from this video, it would be a little bit more clear how CRISPR-Cas9 makes a cut and how it actually disrupts a gene sequence.
Been adapted into a powerful tool for genomic research. There are two components to the system: a DNA cutting protein called Cas9 and an RNA molecule known as the guide RNA. Bound together, they form a complex that can identify and cut specific sections of DNA. First, Cas9 has to locate and bind to a common sequence in the genome called a PAM. Once the PAM is bound, the guide RNA unwinds part of the double helix. The RNA strand is designed to match and bind a particular sequence in the DNA. Once it's found the correct sequence, Cas9 can cut the DNA. Its two nuclease domains each make a nick, leading to a double-strand break. Although the cell will try to repair this break, the fixing process is error-prone and often inadvertently introduces mutations that disable the gene. This makes CRISPR a great tool for knocking out specific genes.
Okay, so here this one is the wrong basis that has been inserted by the NHEJ pathway. So the next sequence is disrupted, so it is deactivated. So okay.
Redacted.
So now this is the end of part one, and I have discussed the basics how CRISPR-Cas9 works to knock out or to deactivate or to mutate a gene or a portion of DNA. Now in this part two, this is the second part of my presentation, and this is the largest part. Other one was a smaller part. So here I will talk about different tools and techniques that has been derived from this system, this amazing system, and I will give you some brilliant example of each of the tools and techniques that I would cover in my later part of presentation. So yeah, so let me start with the gluten problem that I discussed in my introductory slides. So I'm going to give you some few examples of knocking out. So I told you that how gluten is actually causing problem because many people are allergic to gluten, so they develop the celiac disease in order to, if they consume this wheat, rye, and barley, because they have this gliadin protein. Okay, this gliadin is antigenic. But it's not true for all human beings, but some humans, this gliadin is antigenic to them. So if you see the gliadin gene, I told you that one protein is produced from, most of the protein is produced from a single gene. So here, if you see the structure of the gliadin gene, this is the gliadin gene. This is different domains, okay? So now, actually, what part of this gliadin protein actually is antigenic to the human immune system? So that's called epitope. You know, any protein which is considered as a foreign protein or as an antigen, the body develops antibody against it. Okay, so in case of this gluten allergic people, gliadin is also considered as a foreign protein or antigen. So this portion of this gliadin gene, this red highlighted portion, is actually the epitope of this gliadin. So this portion is recognized by the immune system to develop antibody against. So what they have done, a few scientists from Spain and the Daniel Voigt is also from USA, they designed the guide RNA. Guide RNA is the genomic address. So here is the guide RNA, here is another guide RNA they designed, and they make a cut specifically here and here in order to deactivate this gliadin gene. So you can see this is a nice example from a multi-top analysis of gliadin, extracted gliadin from the edited one and the control one. If you see this peak, these black peak actually designate the control one. This is alpha gliadin, and this is the intensity, you know, high amount of gliadin is present in the non-edited one. And these two peaks, this is blue and this yellow peak, this designates the extracted gliadin from the edited one. So you can see the huge reduction in the intensity of signal. So huge reduction of alpha gliadin amount in the edited one. So this kind of low gluten non-transgenic wheat has been developed using CRISPR-Cas9 knockout, and obviously this is this one is the edited one, this one is the edited one, this is the control one, this is the control one. So they all look similar in other phenotypes, but reduced in gluten content. So this is a very classical example how to remove the anti-nutrient factor from our food using CRISPR-Cas9, and that is specifically by activating that gene. Okay, so now if you see another classical examples, and that has come to news, I think all of you probably know because it has been a lot discussed. So is Enon Young from Penn State. So he was my supervisor during my postdoc at Penn State. So what they have done, because you know Pennsylvania is actually the biggest producer of mushroom in USA, and what Enon took interest in that because you know the mushroom, when you cut mushroom and keep it for a longer time, then the cut end becomes brown or black, okay, and people do not like to take it. So These phenomena actually happen due to PPO, the activity of an enzyme that is called polyphenol oxidase. This polyphenol oxidase actually oxidizes phenols and they form this black pigment or brown pigment. So what they have done, Inong's lab has done, knocked out this PPO gene in the agaricus mushroom, and what they have got, the knocked out PPO genes, mushrooms actually showed very less browning compared to the control one. So this is the non-browning mushrooms, and that has already been cleared by USA regulatory body that as it does not contain any foreign gene insertion or anything, so it would not be regulated as GMOs. So this is a classic example of knocking out of an anti-nutrient factor. Now, so this is just I want to touch you on this one. So I told you that in order to reactivate a gene, you have to have an NGG at your target site. The target site should be followed by NGG sequence, but you might have already in your mind that what if I don't have an NGG sequence at my target site? So can I not target my gene, or can I not deactivate the gene? So you know, this field is very rapidly progressing. It's like in a storm, you know, you cannot follow the storm. So every day there are a lot of publications. So there are a lot of development on these lines. So there are a lot of orthologous Cas9 developed or discovered from different bacteria, and they could recognize other PAM, not only NGG PAM. So NGG PAM is recognized by Cas9, that is Streptococcus pyogenes Cas9. But scientists have came up with other orthologous PAM from other species, like I told you, Streptococcus aureus, and then CJ-Cas9, NME-Cas9, STH-Cas9. There are a lot of. So you can see, and also scientists have engineered the original Cas9 by changing certain amino acid in that Cas9 to make it recognize NG instead of NGG. So you can see if you have N and G, one single G, then these are the variants that you could use to target your gene. If you have a target sequence NNG or a T, and then you can use SA-Cas9. Then suppose you don't have a GC rich sequence, so your gene is present in a TA rich sequence. So you don't have that option to use the SP-Cas9 or SA-Cas9 or other kind of Cas9. So you can go for Cas12a. It can recognize the PAM triple TB. So your target and then TTTV. Okay, so it is sufficient to target in order to be targeted by Cas12. Okay, so these are the examples. So yeah, we recently wrote a chapter for one of our upcoming book on CRISPR-Cas methods. So we discussed all of this, and we tabulated all of the different kinds of engineered and orthologous variants of Cas9. So you could go through. Now, another question you might have already have in your mind that, okay, I can deactivate one gene, but in certain biosynthetic pathway, suppose some anti-nutrient biosynthetic pathway, you want to get rid of that anti-nutrient, but it is not possible to get rid of that anti-nutrient factor by deactivating a single gene, but you need to deactivate a whole lot of 3 or 4 or 5 or 6 genes in order to completely deactivate the whole biosynthetic pathway. So what would you do? So this CRISPR-Cas9 give you that flexibility to target multiple genes in a single experiment. You don't have to go for one by one. It's not no need. So you can deactivate multiple genes in one go. So how it acts? So suppose you designed a construct which has a Cas9. So now you have to give 3 different addresses, or 4 different or 5 different addresses. So suppose these are the 3 different guide RNA that actually designate 3 different genes in the genome. So this Cas9 go and take the first address, then Cas9 go and take the second address, and then Cas9 go and take the third address. And then they go to gene 1, gene 2, and gene 3. So all 3 are deactivated. So you do one experiment and you knock out or you deactivate 3 or 4 or 5 genes. So in our lab in the USA, we did 8 different knockout of 8 different genes in one go, in one single experiment. So it is possible, no problem. Now, So now let me give you an example of multiple targeting and multiple deactivating, how it is helping in plant biology and how it is helping in discovering new trait governing genes. So this is a classical example from Zhang and Zhu's lab, recently published in PNAS. So, these are the genes, PYL gene, and PYL has 13 different members. This is a gene family which has a lot of gene members. So now they wanted to investigate which gene is actually more important for one single trait, okay? So what they have done, they designed 8 different guide RNA to deactivate all 13 of the genes, and they went for knocking out all of them, and what they have came out with a triple mutant. So if it is mutated, this PYL1, 4, and 6, if all 3 are mutated, then the phenotype is like that. Here all 3 are mutated, the phenotype is like that, and they also showed higher yield and higher vigor in the plant growth than the control one. So it would not have been possible if it is not a multiple targeting system, So they have got a lot of combinations of 2 mutants, 3 mutants, 4 mutants, 5 mutants, and they analyzed all of the mutants for the phenotypic analysis. And then they got one triple mutant, this is which has deactivated gene 1, gene 4, and gene 6, and they got a better yield than the control one. So this is the beauty of this system. Now, if you see another classical example from, this is from Jack Lipman's group from Cold Spring Harbor Laboratory. So this has been published in Cell. This is a classical example of promoter editing. So what, the promoter, what does it do? Promoter and then gene. So promoter controls the expression of the gene, okay? So how gene is producing, how much RNA, how much protein would be produced from that gene, it is actually controlled by promoter. So what they have done in tomato, they targeted a promoter of a gene with 8 different gRNAs, guide RNAs. So 8 different position has been targeted with different guide RNAs. So it will make 8 different cut in the promoter. So in the repair procedure, it will make a lot of combination. First cut, second cut, third cut, fourth cut, fifth cut, sixth cut, seventh cut, 8 cut. So 8 cut, and this 8 cut makes a lot of different kind of novel 6 regulatory alleles in the promoter. And this kind of combination, when they got, and they will go for large phenotypic screening, and what they have seen surprisingly, that these are the control panel. You can see this is the wild type, and you see it has 6 florets in the control tomato flower, whereas in the edited one it has 9 florets. So the floret number has been increased, and you can see the size of the fruits. It has only 3 locules in the control wild type one. After this promoter edited line, 1 or 2 lines, they got 11 locules with huge size of the tomato. So this is also a classical example of multiple targeting and multiple cutting in the regulatory region or in the promoter, and how it could help in developing novel kind of allele, and it could be translated into novel phenotypes that would not have been possible through normal transgenics or normal traditional breeding. So now, if you see, okay, so now this is exciting. So now I'm going a little bit farther from this normal knocking out kind of stuff. So if you see beyond double strand break in the genome, so I told you about how Cas9 actually causes the double strand break, and no need to discuss again. And this one is called Cas9 or Cas9. So scientists have engineered the Cas9 by changing a single amino acid in the whole Cas9 protein, and it makes a single strand cutting protein, not a double strand cutting protein. So you just take out 1 scissor from the postman and you make the Cas9 as a Cas9. So it can cause single strand break instead of double strand break. Now similarly, you could catalytically deactivate the Cas9 to make it as a dead Cas9. So I told you that Cas9 has 2 properties, 2 principal properties. One is to bind to a perfect sequence that is guided by the guide RNA. and then it makes a cut. So it has two principal properties. So now what I am talking about here, the cutting property. So this dead Cas9, it means it is dead in sense of cutting. It could not, it would not be able to make a cut in the sequence, but it has the ability to bind. Where to bind? The guide RNA. The guide RNA actually directs its where to bind. It will go there and it will bind, but it cannot bind, it cannot make a cut because this Cas9 is made a dead Cas9 by removing both the scissor. This has been done by by two different mutations. Okay, so this is called dead Cas9. So why I'm telling you all of this, because I am going to introduce you to the tools where this kind of naked Cas9 and dead Cas9 is necessary. This is fascinating. So if you see the gene regulation, playing with gene regulation, this is CRISPR-I. So this is called CRISPR interference. We know that RNA-I, RNA interference, this is kind of CRISPR-I, CRISPR interference. So here, this is dCas9. Now this dCas9 is deactivated Cas9 and it is fused with CRAB. CRAB is a transcription inhibitor. So this dCas9, you give this dCas9 with the guide RNA. Guide RNA is the address. It actually designates where to bind. So guide RNA takes the dCas9 to bind to the promoter or enhancer. And now the dCas9 is fused with another repressor protein, that is CRAB protein. So now actually what dCas9 is doing here, dCas9 is actually ferrying, it is acting as a ferry of a protein from one place to another place. So when dCas9 brings CRAB to promoter or enhancer, then this portion is blocked. So the CRAB inhibits the transcription of the gene. So this is how it could help in deregulating or down-regulating any gene of interest, if you want. Now, this was the example for animal system, but in plant system, this CRAB does not work very well. But what they have came up, this is another protein, SRDX. This domain has been fused with dCas9, and it seems very good in inhibiting plant genes. So this is another example. Now, if you see another, likewise, you could make a CRISPR-A tools, that is CRISPR activator. So you could increase the expression of certain genes by using CRISPR activator. So here it is called transcription activation. So here same, dCas9 guide RNA is fused with VP64. This is viral protein 64. This is, so I told you the dCas9 can bring this VP64 to the target. So here target is the promoter, where dCas9 comes and bind, and it brings VP64 along with it because it is fused with that protein. So VP64 is a known activator of transcription. So when VP64 is present specifically at that promoter, so that gene would be expressed in a higher level. Okay, so this is how transcription activation works. So similarly for plant system, this VP64 does not work very well. So that's why another group, they have developed TV, that is fusion of VP128 and another protein, tail protein, and they make an activator with fusing with dCas9. So you can see here the expression. This is the wild type expression, and this is the expression where you have treated with dCas9 and TV. So this is how you could increase the expression of certain genes in any of your species of interest. Okay, so now this is another fascinating area, that is epigenome editing. So epigenome editing, epigenome is nothing but something which is controlling expression but does not depend on sequence. So how? Because in the same gene is present in one variety, the same gene is present in another variety, but that gene, in one variety the gene is expressed very in higher amount, in another variety the gene is not expressed that well. Why this is so? In many cases this is because the promoter of that gene is methylated. So methylation actually causes, sometimes methylation causes the reduction of expression of that gene. So here if you see, suppose in this example, the dCas9 is fused with a demethylating enzyme, that is TATE1. So this TATE1, this dCas9 brings the TATE1 to the promoter which you know that it is methylated and it is not expressing well. So you want to get rid of that methylation, okay? So you don't want to change the promoter sequence, but you want to get rid of that methylation. So you fuse dCas9 with the TET1 and you give a guide that will take the dCas9 and TET1 to that promoter. So you go to the promoter and this TET1 function here, and it will demethylate. And after this demethylation, the gene become more expressed than the wild type one. So this is how you could edit the epigenome signature of any organism with this CRISPR tools. So now this is a classical example from Arabidopsis. So this is the FWA mutant where it has delayed flowering, because of the demethylation, but when it is methylated, it shows early flowering. So you see, this is an example of using the epigenome editor. If it is methylated, then it shows early flowering, whether the wild type did not show that kind of flowering at that stage. So this is a classical example. So now here, this is another interesting area, live cell chromatin imaging. So you could visualize where is your DNA present in the cellular scenario, and how what are the processes going on there, and many kind of things you could do with this beautiful tool that is called chromatin imaging. So you could image RNA, you could image your gene of interest where it is present at certain stage of life cycle. So this is a classical example of here. So I would give you an example here. So these, in order to understand, this dCas9 is fused with a fluorescent protein, and I'll give you and give them a guide RNA addressed to your gene of interest. So it will make a complex and it will go to your gene and it will bind there. You can visualize under the camera that, oh, dCas9 goes and bind with my gene of interest, so the fluorescent will come from that portion, and you would be knowing that this is my gene. So this is how chromatin imaging works, and this is a classical example. There is a small video. This was a recently published paper in Science just few days ago. So here you can see this green is actually the green fluorescent protein fused with the Cas9, active Cas9, not the dCas9 one. And this one, the red is another fluorescent protein that was fused with repair protein, double strand break repair. So how fast it happens after it makes a cut, how repair protein comes very quickly to repair that double strand break, you would understand from this video. So you see, this is the portion where Cas9 making a cut, and now the red protein is coming. Red protein is a repair protein. Okay, it is coming that in that locus to repair it. So it is very fast. So how it happens? So it is very interesting that after the Cas9 makes a cut there, immediately other repair protein comes there to that loci exactly, and they try to repair that break. Okay, so now this is another interesting area, that is CRISPR detection. So CRISPR is now being utilized to detect the presence of pathogen, to detect any kind of DNA or RNA in the sample. So this is called CRISPR detection, a new area which is emerging area. So we all know that we are going through a pandemic, and this pandemic is due to the COVID-19 or SARS-CoV-2 virus. And the early asymptomatic patient is very difficult to detect. And also in case of any kind of viral infection, if you have suppose one copy of virus in 1 microliter of blood, it's very rarely you could detect that this sample is positive. So now CRISPR makes a very, very highly resolutable detection system. So if you see, there is a detection system detector developed by Jennifer Doudna's lab. So how it works, you can say, suppose this is a protein Cas12a, and this Cas12a has a special activity which I have not told you till now. This is called collateral activity. Collateral activity means it will go and bind to its target DNA, obviously, and it will make a cut at the target DNA. But along with cutting the target DNA, it can also cut the adjacent DNA. This has a special property of Cas12a protein. So Cas12a protein, you give a guide RNA, and now this guide RNA is designed to bind with a viral DNA, suppose viral DNA, okay? Now, when you give it to, suppose you have a tube that has a viral DNA, you don't know whether it has or not. So now Cas12a guide RNA, and with the sample you add that. So Cas12a guide RNA binds with the target DNA if it is present in the sample, and it makes a cut. Obviously, this is the normal mechanism that you understand already. Now you have to add another thing in that tube, that is reporter coencer dye combination. So this blue is a coencer and this yellow is a reporter dye. So these two molecules, this reporter dye can fluoresce, can show fluorescence if it is not present with the coencer one, because this coencer molecule actually absorbs all the fluorescence from it. So if they are in combined position, so this fluorescence cannot be detectable. But if it is if they are separate, then the fluorescence will be detected. So you see this coencer and then small DNA, any DNA, and then the reporter. So if you add that kind of molecule here, so I told you that Cas12a go to bind the DNA target if it is present in that tube. So if it finds, suppose that viral DNA, it will bind there, it will make a cut at the viral DNA, and it will also make a cut at the DNA which is present, which was used to link the coencer and reporter. So when it is separated, then the reporter will fluoresce, and you can detect it by lateral flow, strip, or any other mechanisms. So you would be understanding, yes, the target DNA, the target viral DNA is present in that tube. That's why I got this fluorescence. Okay. So now similarly, another one is Cas13. So Cas13 is a very nice Cas protein that has a different property that could bind to RNA and cut the RNA, not the DNA. As my presentation, I told you that Cas protein binds specific DNA and then make a cut. But this Cas13 has a unique property to bind RNA and make a cut in that RNA. So similar mechanism or similar a similar idea has been used to develop another detection system that is called Sherlock. It is developed from MIT Harvard, Feng Zhang, by using Cas13. So, you know, COVID-19 is an RNA virus. So you can use to detect that COVID-19 with the RNA, to detect the COVID-19 using this Sherlock system, okay? But you may ask me, how detector could not be used to detect the COVID-19? Yes, you could do. So then you would ask me, no, COVID-19 is a RNA virus. How could you use a protein which could detect DNA? So the question is, in order to answer that, so you have to isolate the RNA, and then you have to reverse transcribe to make a DNA, and then you can use the detector, okay? Now, so if I want to give, this is a case study. So this is a recent paper. This is based on Cas12. It means it is based on detector system, and this is a stop COVID workflow. This is based on Sherlock system, and it has already been approved by the US agency to conduct mass testing for COVID-19, especially in USA. And now if you come to the plant, because I'm talking about plant biotechnology innovation, so I should always give you some example of plant. So this is, you know, this is from Feng Zhang's lab. So nucleic acid detection of plant genes using CRISPR-Cas13. So Sherlock system has been used for plant detection. This is another from Kevin G. Kevin G was a former postdoc of our lab. So he was my senior in that lab. So evaluation of CRISPR-Cas12 based DNA detection for first pathogen diagnosis and GMO test in rice. So this has already been used, and it could be used to detect any kind of presence of trait or pest surveillance, and also GMO detection, whether suppose we are importing some foreign seeds, or we are exporting some foreign seeds, so it could be used to detect whether it is a GMO seeds or not. So it could be used that. And also, more importantly, it is very, very useful to detect any pathogen like bacterial pathogen, fungal pathogen, or viral pathogen. Okay, so now I'm going to a very next step of my presentation, that is actually precise editing. I will be talking about precise editing briefly. Probably I'm going out of time. Yeah, so this is an example, very classical example of how single mutation could affect or could cause a genetic disease. So this is a snapshot from a movie of Amitabh Bachchan that is called Pa. So here is Oro. Oro is suffering from progeria, and so progeria causes rapid aging at the childhood. So the child looks like a very aged person at the age of 12 or 13 years. So here, you know, what is the difference between their genome? So in one gene, these Oro's friend has a G, but Oro has a mistakenly A. So this single difference actually caused Oro to suffer from this rare genetic disease. So similar in case of plant, this is an example of a plant and a mutant. So this is affected with blast pathogen. So you see, this left panel is actually the gene has a C, whether the right panel has a T. So because of the C and T difference, it is highly affected and it is less affected. So this is how single changes in the DNA later could change hugely in the impact. So the question is, do we have a tool, or do we have a way to rewrite specifically or rewrite precisely the genetic letter? If we have a Oro's A, can we make it a G? If we have a C in the key tag, can we make it as a T to make it a resistant one? The answer is yes, after the innovation of this base editor. So there are two kinds of base editor. One is adenine base editor that could change A letter to a G letter. Another is cytosine base editor that could change a C letter to a T letter. So this has been, actually invented from two labs. One is from David Liu and another is from a Japanese lab. So here is two base editor. Okay, so let me just share this video with you to make you understand.
Then be used to transport those enzymes to a specific DNA sequence. In one example, Cas9 is fused to an enzyme, a deaminase, which mutates specific DNA bases, eventually replacing cytidine with thymidine. This kind of precise gene editing means you could turn a disease-causing mutation into a healthy version of the gene.
Okay, so it causes specific, okay, so I will explain how it works actually. So this is adenine base pair, so how it has been developed. If you see the structure, this is an architecture. So ANGRAU has been used here. Now I talked about normal Cas9 and then dCas9. Now this is the ANGRAU. So Cas9 has been used here and fused with TAD-A. TAD-A is a deaminase enzyme that is fused with the ANGRAU to make it as a adenine base editor that could change A to G in the top strand and T to C in the bottom strand. So, okay, so how it actually works, if you see, suppose you have a defective gene that is AT. Suppose in case of Oro's gene is defected because of the presence of these A. Now you want to make, you want to correct it to a GC one. So you have to apply ABE. So adenine base editor. Now when you apply adenine base editor, so adenine base editor will go to that locus and it will deaminate adenine, because I told you that ABE is made up of ANGRAU fused with adenine deaminase. So the adenine deaminase portion deaminate adenine to make it an inosine. Okay, so now you see IT is not usual. It is not a perfect base pairing, so it is a mismatch. Now cellular mismatch repair immediately come into action to remove one of the bases, but cellular mismatch repair would not understand which one is the actual mismatch. So in order to trick the cellular mismatch repair mechanism or machinery, so the ANGRAU will make a single cut or nick at the T containing strand. So when it makes a cut here, now cellular repair machineries, they will take or they will consider these broken strand as the damaged one. So it is common to consider this strand as the error strand, because they see that this has a nick, so it should be the wrong one. So cellular repair machinery, they remove the T. They remove the T. They don't remove the I because of the presence of nick. So when they remove the T, then they insert a C opposite to I, because I is always read as guanine in the cellular system. So opposite to I, C is incorporated, and in the next step it becomes a GC base pair. So now you got a defective gene corrected to GC with ABE. So I would give you a very good example from my work, how I was able to change 2 single nucleotide to a correct one. Okay, so this is the cytosine base editor, how it works. So let me give you a simple mechanistic understanding. So you have a defective gene CG, you want to make it as a TA. Now you apply CBE. Now CBE is made up of cytosine base, means CRISPR-Cas9 with the.
Can you
Yeah.
take 5 more minutes to
Okay, sure.
go ahead?
Okay, sure. Thank you.
Thank you. Thank you.
Okay, so okay, I'll come later about this mechanism, maybe later, because I'm short of time. Okay, so let me give you an example. So I targeted 2 genes, WSL5 and Z3, and this is a system called PTG system, where 2 guide RNA is separated and they go to each of their targets, this WSL5 in chromosome 4 and this Z3 to chromosome 3. So if you see, this is my gene WSL5, where I targeted this A to change it to a G later, not the other one. Okay, so when I applied adenine base editor, I could change this adenine to a specifically a G. You can see this green peak has been changed to a G, so A has been changed to a G specifically, and we could find the phenotype that is expected due to the single nucleotide change. Similarly, in case of Z3, we are able to change specifically the A to a G clearly. So this is the control one, this is the mutant because of the base editor treatment. Okay, and we got the mutant phenotype here. So we could get the base editor free non-transgenic mutant in T1 generation. So this is the absence of the base editor. If you see the absence of any band, so this is the absence of base editor, but these are all mutant. So we got in T1, we got mutant plant, targeted mutant, but no presence of any base editor construct. So you got non-transgenic mutant plant here. By simple virtue of simple sexual segregation, Mendelian segregation, you know, because the base editor is integrated in one portion of the genome, but your target is another portion of the genome. So in the next generation, base editor would be segregated out, and you select for only the mutant one, not for the base editor one. Okay, so this is another example. This is unpublished data. So what I targeted in one gene that was not producing the full length of protein because of the presence of a premature stop codon, and because of the short protein production from this gene, it is susceptible to a disease. So TAG is a stop codon, you know, this is a stop codon. So translation would be stopped here, and it is not going to produce the full length protein. So now we want to mutate this stop codon to a the coding codon. So TAG became TGG after we applied with adenine base editor, and the mutant now produces the whole length of protein, so it would give you the disease resistance phenotype. Okay, so it has been applied for different kind of applications in plants, so you could change all kind of SNPs for different applications. So now if you go to the HDR, this is the last part of my presentation, so I check maybe 3 or 4 minutes more. So I told you that HDR, how it happens, but it is very extremely low efficiency. We have tried a lot of different, this is also unpublished data from my earlier experiments. So we have tried in tobacco and rice to improve the HDR efficiency, but we have seen very low efficiency in HDR efficiency in plant. Because suppose you want to change different kind of, suppose 3 base pair deletion you want to do exactly, 4 base pair insertion you want to do, you cannot do exactly with CRISPR-Cas9 knockout or base editing, but you have to go for the HDR, but HDR is very low in efficiency. So how to do that kind of precise change? So here Okay, so what I told you, okay, just skip this one. Let's go to the prime editing to avoid this kind of low efficiency HDL mechanism. So this is a new developed editing technology. Can you recognize this two one? This is a red blood cell, this is normal, and this is a sickle cell, okay? And this is because of A2T mutation in beta globin gene, okay? So A2T mutation cannot be done with base editing. So you have to go for HDR, but HDR is very low. So here comes prime editing. So prime editing is a newly developed technology where ANGRAU is fused with reverse transcriptase enzyme, and you get an extended guide RNA. That guide RNA is called prime editing guide RNA, which has here you have to add the sequence that you want to edit, and then you have to add a binding region. This is called primer binding site. So I will explain a bit later. So this is the normal conventional guide RNA, but at the 3' end, you have to add these two extra sequence with the guide RNA. Okay, so here this is the 5' end bind with the target sequence, and the 3' end is extended with the extra sequence that you want to incorporate. And this is Cas9, so it makes a cut here. Okay, now how it happens? So after it hybridizes with the prime binding site, so the 3' end has a primer binding site. It will exactly bind with the nigged strand. And now this red one is the changes that you want to make, okay? And this is called RT template. Now, the reverse transcriptase is fused with the ANGRAU, as told you. The reverse transcriptase will start synthesizing from here, and it will fill this up in the genomic DNA. And now you got this kind of situation where you have a 3' flap, which has the edit. The red one is edit. But in cellular system, you have two flaps. One is 3' flap, one is 5' flap. 5' flap does not have edit, but 3' flap has the red edit. Now in cellular system, they have flap endonuclease enzyme that prefers to cut the 5' flap, not the 3' flap. So now the 3' flap will rejoin with the genomic sequences by ligation, and you got the edited DNA. This is how prime editing works. So by prime editing, you could change all kind of base changes, all 12 kind of base changes. That was not possible through base editing, because base editing could do only transition mutations. But here you could do all kind of transversal mutations also. You could insert 1 base pair to 44 base pair, and it could delete. They have presented in that Nature paper that 1 base pair to 80 base pair could be deleted precisely by using this kind of prime editing. If you see, in a very short period of time, almost 6 to 7 papers has been published in plant system, especially in rice, and 1 paper in wheat. But what I have seen from those papers, that in case of plant system, prime editing efficiency is not that great as it is for mammalian system. So we have to improve the efficiency in order to use prime editing as a common tool for editing precisely in our gene of interest to make a new allele, to delete a bad allele. Okay, so yeah, this is the last few slides of my presentation in order to know more. So what I talked about, Gebra3 and WSL5 changing single base pair with adenine base editor, we have published it in a journal. It is available, freely accessible. You could go through that. And then in chapter 1 of this upcoming book, probably it is going to be published in 2 to 3 days. In chapter 1, we have talked about a lot of different tools, and we have tabulated them. In chapter 4, we have wrote a protocol on how to use base editing in rice for multiple loci editing. So this is experimental protocol. So like all kind of materials, methods, and all protocols kind of things. And this book contains protocols on 11 different organisms, and it's a great book. We have edited it. It is going to be published soon. This is a blog we have written for AdGene for how this plant-based editor. So it is written in a layman language. You could go through this one to understand initially, and then this to review there. For one is extensive on CRISPR-Cas mediated base editor. Another is how to predict CRISPR-Cas9 induced mutations outcome. So this is on machine learning and all these things. So in conclusion, what I could say that CRISPR-Cas has unprecedented ability to generate targeted and specific mutations. Obviously, you have understood that. Now, it is possible to deactivate one or multiple genes in one single go, and it could be also possible to upregulate or downregulate or epigenome edit with the CRISPR tools. And then you could use CRISPR tools for early pathogen detection, what kind of pathogen it is, and then go for precise modifications that are also possible due to the discovery of base editors and prime editing, and then HDR. And then I have not talked about it, I skipped that slide, but you could do a targeted insertion of transgene. So, you know, the opponent of GMO system or transgenic technology say that why the transgene is getting integrated, we don't know, because it is not controllable. But if you use CRISPR-Cas9, mediated insertion of transgene, you could insert a transgene of interest in a safe harbor locus of the genome, because the portion of genome that is known that if it contains any foreign gene, it does not hamper the whole scenario of the genome. So this is for safe harbor locus. So you could targetedly insert transgene to that position using CRISPR-Cas. So in
Thank you.
Thank you very much.
Thank you, Moraji.
Yeah. Thank you for giving me chance for extra time. Yeah.
Very nice, very nice. It is a comprehensive presentation with well designated slides. Since we are running out of time, there are some questions. Few of them which are relevant, I have marked them. I will put it to you.
Yeah.
Some of these questions were answered while your presentation was going on.
Yeah.
Now I find, but the first one is, what is the need of using CRISPR-Cas9 in agriculture? One of the slides in the agriculture you have presented. I take the person who made that question to go through the presentation. Then one question is it possible to produce transgenic tomato leaf curl virus?
Okay. So transgenic tomato against leaf curl virus, am I right? So there are reports, I know there are two reports using Cas protein to develop an artificial immune system to resist against leaf curl virus. Suppose that virus has the code protein. So what they have done, they have expressed the Cas protein with the guide RNA. That guide RNA actually could bind with the DNA or genetic material that is coming from the virus, because we know that virus, when they infect, they inject their genetic material into the cell. So whenever genetic material comes, the cell has the Cas